Developing a single biopharmaceutical product takes between 10 and 15 years. Over the course of that development cycle, nearly 90% of all drug candidates fail before they ever reach the market. For the fraction of products that survive, the estimated research and development costs from initial discovery to product launch reach up to $2.8 billion. But the science is only one phase of the operation. The ultimate bottleneck is navigating a labyrinth of global health authorities that hold absolute power over commercial licensure. A molecule can demonstrate clinical efficacy in a lab setting, but that data is legally meaningless until it complies with the specific legal and administrative mandates of individual nations. In the United States, clinical trial applications must be filed under Investigational New Drug Designations, or INDs. Final commercial licensure requires a biologics license application for large molecules or a new drug application for traditional prescriptions. Cross into the European Union, and the terminology shifts. The European Medicines Agency demands clinical trial applications, followed by a centralized marketing authorization application. Discovering a breakthrough medical treatment solves the biological problem. Engineering its survival through overlapping global regulatory systems solves the distribution problem. Executing a worldwide rollout requires physical distribution across entirely different legal ecosystems. Manufacturers must submit documentation to over 80 independent health authorities, each enforcing its own specific chemistry, manufacturing, and clinical guidelines. To manage this, pharmaceutical companies build what is known as a core dossier. This is a master file of scientific evidence designed to be leveraged across multiple international markets simultaneously. Constructing entirely separate, highly customized submissions from scratch for every single country would introduce severe operational delays and drive up authoring costs to unsustainable levels. Many smaller or developing markets alleviate their own bureaucratic workload by relying on a reference model. They require the manufacturer to secure initial approvals from reference markets like the US FDA or the European EMA before granting local clearance. This creates a direct conflict between the manufacturer's need for standardized global harmonization and the strict enforcement of localized regulatory sovereignty. To navigate this fragmented landscape without duplicating decades of laboratory data for every border crossed, the industry needed a standardized architecture for its evidence. The solution emerged from the International Council for Harmonization, or ICH. The ICH operates as a consensus-driven body with a clear mission to create a highly resource-efficient, standardized framework that regulators and industry experts can use worldwide to register safe medicines. Their primary architectural achievement is the Common Technical Document. The Common Technical Document arranges pharmaceutical data into a 5-module pyramid. Modules 2, 4, and 5 form the universally accepted scientific argument, comprehensive summaries, toxicology, and clinical efficacy. Module 3 contains chemistry, manufacturing, and control data. Module 1 houses unharmonized regional administrative information. Module 1 contains country-specific application forms, localized product labels, and promotional materials. These documents are tied to national laws and cannot be universally standardized. The CTD successfully aligns the core scientific evidence across global borders, but it hits a hard limit when it encounters sovereign administrative boundaries. Even with a standardized digital dossier, manufacturers face legacy regulatory friction. Advances in manufacturing technology often outpace outdated national laws. For decades, regulatory standards mandated the General Safety Test and the Abnormal Toxicity Test. These required manufacturers to inject specific volumes of product batches into guinea pigs and mice, observing them for a week to check for extraneous contaminants. The US FDA removed the General Safety Test from its Code of Federal Regulations in 2015, determining it was duplicative and no longer necessary. The EU removed the Abnormal Toxicity Test in 2017 to align with modern mandates reducing animal testing. However, health authorities in several emerging commercial markets still require these tests to be performed locally before a product can clear customs. In-country testing mandates force pharmaceutical companies to physically ship samples of every intended commercial lot to government laboratories for independent verification against the company's own certificates of analyses. In China, for example, the National Medical Products Administration mandates strict in-country testing for biologic drug substances and commercial vaccines, with no formal process for transferring the manufacturer's own test methods to the state institute. Turkey enforces a different physical barrier. Their Ministry of Health requires a complete good manufacturing practice pre-clearance, necessitating either an on-site or paper inspection of every specific manufacturing facility before a commercial application can even be submitted. These localized mandates severely complicate the post-approval supply chain. Extending a product's shelf life or increasing batch size requires individual review from every health board, a process that can take anywhere from a few months to over 2 years, depending on the country. A perfectly organized electronic dossier cannot bypass a physical testing mandate or a manual factory inspection required by sovereign law. Vaccines carry an entirely different set of regulatory behaviors compared to standard pharmaceuticals. The guiding axiom for biologicals is simple: the process is the product. Every vaccine manufacturing run is highly unique, and the concept of a fully standardized, identical molecule does not apply. Because of this inherent biological variability, both the United States and the European Union maintain strict mandates, requiring lot-by-lot release testing executed by government laboratories prior to commercial distribution. This level of continuous physical government verification is no longer required for most traditional small molecule drugs, which can be validated entirely through analytical chemistry. The complexities of organic material require even the most highly harmonized regions to demand constant localized physical verification for every single batch produced. In 2020, the COVID-19 pandemic subjected this rigid global apparatus to extreme stress, forcing regulators to evaluate vaccines at a speed previously thought impossible. This chart shows the timeline of a traditional clinical testing model. Drug development is heavily sequential. Researchers must fully complete Phase 1 trials, compile the safety data, submit it to the regulator, and wait for approval before Phase 2 efficacy testing can even begin. To deliver a viable vaccine candidate in under a year, companies had to implement a parallel clinical trial model. Looking at Pfizer's 2020 development strategy, the sequential steps were broken apart and stacked vertically, compressing a standard 10-year projection into 12 months. They simultaneously tested 4 different vaccine candidates at 3 distinct dosage levels, recruiting over 46,000 patients across 153 clinical sites in 6 countries. To achieve this, the manufacturer had to build the necessary infrastructure and manufacture millions of doses at risk, absorbing an estimated $2 billion in operating costs before the clinical data had proven the vaccine was actually effective. By running Phase 2 and Phase 3 trials simultaneously, a testing progression that typically takes a full year under normal circumstances was executed in roughly a month. Compressing the schedule required stacking regulatory requirements and absorbing billions in potential losses while adhering to the same safety metrics. Accelerating a clinical trial provides zero public benefit if the health authorities are still operating on a 12-month review cycle. Regulators activated specific review mechanisms to match the speed of the science. In the United States, the FDA activated the Emergency Use Authorization, or EUA. The EUA allowed for rolling data submissions. Instead of waiting for a fully completed dossier, manufacturers could submit clinical data as it arrived. The FDA also established a baseline standard early on, requiring a placebo-controlled trial to demonstrate at least 50% efficacy for consideration. Concurrently, the European Medicines Agency offered a conditional marketing authorization pathway. The logic of the CMA rests on a calculated risk-benefit analysis. The agency grants temporary use based on less comprehensive initial data, determining that the immediate availability of the vaccine to the public outweighs the inherent risk of an incomplete dataset. On a global scale, the World Health Organization utilized an emergency use listing procedure to expedite assessments for unlicensed vaccines and diagnostics in developing nations. The WHO guidance allowed manufacturers to substitute traditional, drawn-out process validation with continuous process verification, approving the initial commercial production while data collection continued post-launch. These overlapping regulatory flexibilities allowed the clinical science to reach distribution. On December 11, 2020, the physical result of this parallel system achieved authorization.
The pandemic response demonstrated that while the global regulatory apparatus is fragmented and complex, it contains the administrative agility required to accelerate pharmaceutical distribution when aligned against a singular threat.